Hey, ladies and gents, welcome back to More Biology. We're going to finish up our central dogma. So we started with 8.2 with DNA. We then we learned how that replicated during mitosis, during the synthesis phase. And then in 8.4, we did transcription, where we took that message from our DNA and we put it into the language of RNA. And now we're going to actually take that message of RNA and we're going to make it into the protein that we need. All right? Are you ready? I know I am. Let's get started. All right. So translation, remember, takes place. We have the rRNA, right? The rRNA, and that is on the ribosomal RNA. You have the big subunit and the small subunit. And since it's ribosomes, we're seeing a lot of this on the rough ER. Okay, so you will see this out in the cytoplasm where all of these ribosomes are taking place. All right, so the structures that are involved in the translation, again, remember we have the mRNA, and this is from, it's taking the message of the DNA. So we get this inside the nucleus. It goes outside the nucleus into the cytoplasm. Then we have the tRNA. This is the transfer RNA, and this is what they call the three-leaf clover shape. Okay, I don't think I'm going to have you be able to draw this, but you'll need to be able to recognize it. Okay, this right here, the very bottom one, this is where the anticodon is. Okay, and up here is where the amino acid attaches, okay? And this is the ribosome, or this is the rRNA. And you have the big subunit and the small subunit. Okay? And then you know the amino acids are free-floating. These are free-floating. Let me go back. Free-floating. Hey, boo! Thank you. Just got my IB test back. Woohoo! Grading for me tonight. Free-floating amino acids in the cytoplasm so that the tRNA can attach, release it, making the amino acid with protein chain, go back in the cytoplasm, reattach, and keep building the chain. All right. So the rRNA, the ribosome. Okay, so you have, like I said, the large subunit and the small subunit. What I think of this is like if you have a piece of paper, your hands are like the RNA. You have the big subunit and the small subunit. And the mRNA, it's hard to do by myself, comes along and it reads it and it moves along the mRNA and it's reading it in the codons as it goes through. That's how I think of it. So it's just like, right? And as it's reading it, it's building this amino acid chains to become a protein. But that's what I think of is the ribosomes as your hands. And it's the big subunit or the large subunit and the small subunit. Okay? The tRNA, like I said, is the transfer RNA. And this is the one that is carrying the amino acid. So like I said, this is the anticodon. Okay? Oops, no. This is where it attaches the attachment point. This is where the amino acid is attached, okay? So the anticodon, what does anti mean? The opposite, right? So if it's anticodon, it's the opposite of what the codon is. And we will practice this, okay? All right. Oops. Here we go. You got the anticodon is the site of the 3-base sequence that recognizes and matches up with the codon of mRNA. So if our mRNA is, let's say, GUC, right? This is our mRNA. Our anticodon, our tRNA, so what's the opposite? C, U, so that would be A, C, G. So this would be our anticodon. Okay, so it's just the opposite of what the mRNA is. Okay, so what's happening here? All right, so the first thing that we need to have is we need to have in the mRNA, AUG, right? That is the start codon. And that start codon is what tells us that this needs to happen. Okay. Then you have the first attachment site. Okay. The large subunit comes in. Look at this. Isn't this neat? Comes in. And then we'll attach this and start the... Start with that first amino acid methionine. Okay. Then another one. So then that's the first one. So then we have AUG, ACG. So that would be UGC. Look, this comes in. So this one will come in. Drop its THR. THR will be here. This will come back out. This moves over, and then we keep reading it down the line. Excuse me. Phew. All right, so let's talk about the elongation part. So remember, between these amino acids is a bond, and this is a peptide bond, okay? So it's coming down, we're growing the polypeptide, we're reading it. As this gets unloaded, it comes back into the cytoplasm, finds the same, the correct amino acid, comes in, and it can reattach the amino acid if necessary, okay? Now there are different sites of the, here we go. Look at that. Isn't that neat? Let's see that again. Nice, huh? All right. So when we stop, when we are at when the gene no longer needs to be coded for, we have something called a stop codon. Okay, a stop codon. And if you go back to your mRNA reading list, there are 3 of them. I know one that I remember. UAG is a stop codon. Just so when it gets to UGA, it knows that it won't code for anything, and that this can get released and the protein can go into the body wherever it needs to go. Okay, so then it falls off. The release factor, look at that. The subunits come apart, they're free-floating in the cytoplasm. The protein goes to where it needs in the body, and the tRNAs get the amino acids ready for the next gene that needs, or the next protein that needs to be translated. Okay, oops. Now in the ribosome, there are different active sites. So this is where, when you go, if you go back and you look, well, at the P site, this is what's happening. So you have APE, okay? APE, APE. And so the A part is where it binds, okay? The P part is where this holds the growing polypeptide, and you can remember P, polypeptide, and E is for exit, and that's where the tRNA goes off, and it joins in the cytoplasm and gets another amino acid. So here, holds amino acid to be added, so this is where it gets added, binds. This is where it holds the growing polypeptide chain, the PP, and E is for exit, where the tRNA exits it out and goes and picks up another one. Okay. So here's just an overview of, the whole process of translation. So you have the subunits, you have the the APE site, right? And the subunit, it comes in, you have to have the start codon. It starts reading it, 1, 2, 3. The anticodon has to match it, so UUU would be AAA. Once this gets binded, the amino acid bonds with a peptide bond to the chain. This goes back into the cytoplasm collects another free-floating amino acid and can come in and bond the amino acid if it's needed again. Okay. At the end here, we have this long chain, polypeptide chain. When it's done, you get to a stop codon. That stop codon needs a release factor. That release factor comes in when it reads the stop codon, releases the ribosome and the protein to go throughout to the cell or where in the body the protein needs to go. All right, I hope that helps. We're going to do this in class, so this is the whole overview, but we're going to do that in class. Okay. We'll go through that. We'll go through this as well. Okay. But this is, you can hit pause at the end like here and see things. But we're going to do a little practice. Okay. So here we have our DNA. Okay, let's go A, T, T, C, G, A, C, G, A. Okay. 1, 2, 3, 1, 2, 3. Oh, that's perfect. So now let's do a replication. DNA replication. Okay, so you're replicating, so this would be T, A, G, C, T, G, C, T. Okay. Now let's do mRNA. RNA. So A. Now you were going to say T, weren't you? No, because T becomes a U, C, G, A, C, G, A. Okay. And then now we have to read our mRNA code. So remember, these are in codons. Let's go here. So read it in 3, read it in 3, read it in 3. So AUU would be AUU ISO. Oh, nuts. Okay, you got the picture, hopefully. I'm not going to start over because this is a long video. But that's what you're going to have to do for the test. And we'll practice that in class, so don't freak out. And it won't disappear on my screen when I hit Control Natilda. Okay? So yeah, so that is just what the whole process is from DNA replication, transcription, and translation. I know you're going to have lots of questions, so come with them ready in class. All right, have a good day, and I will see you in the lab.